Welcome to Kelvin's PhD completion seminar. It's a pleasure to introduce Kelvin Yip, who started his PhD studies with me 3 years ago in 2011. Kelvin was a graduate from the Chinese University of Hong Kong and wrote asking whether there might be a PhD position. And so he flew to Melbourne to interview, and it was clear that Kelvin wanted to study the interactions between the niche cells and cells at the base of the crypt in epithelium that would react to the niche. And I hadn't really studied niche very much, and I was feeling that it was a little bit off topic, but Kelvin convinced me that it was useful for us to study if we wanted to understand crypt biology. And as a result of that, Kelvin has really influenced a number of the experiments we do in the lab, and you'll see the progress that he's made from really an idea to real experimentation. So, Kelvin, welcome to the podium.
Good afternoon, everyone. Today, I would like to share my PhD project with you, which is characterization of the niche regulators that stimulates the formation and maintenance of colon crypts in vitro. As you see, this diagram here is an ex vivo colon epithelium where we culture in a 3D culture called organoids. And by using this excellent cell model, we are able to study how the niche factor regulates the crypt formation in vitro. And I will tell you more about this in a minute. Here's the outline of my presentation. First, I will tell you the research question, and then a bit background on the small intestine and colon, intestinal epithelial stem cells and niche, and the cell and some possible cell model that we can use to study intestinal epithelial cells in vitro. My research question is, what are the mesenchymal factors regulating the biology of colon epithelial cells? And we have two aims in this project. The first is to examine the response of colon epithelial cells to the factors and to identify what are these factors. Here shows the small intestine and colon. And we have a closer look on the intestinal mucosa. We can see the epithelial cells, this single layer of epithelial cells forming crypts and finger-like projection called villi. However, in colon, there's no villi, but only crypts. Let's look at the intestinal crypts. In small intestine, you can see at the bottom of the crypts, there are two populations of stem cells called crypt-based columnar cells. They are labeled by LGL5 and ASCL2. And there's the other population of stem cell called +4 cells, where they locate at the +4 position of the crypt. And in small intestine, there are Paneth cells labeled in blue here, where you can't find in colon. These stem cells proliferate and differentiate from progenitor cells, and then they migrate up to form mature cells. However, when there's a mutation in APC, there will be constitutive active Wnt signaling, and it will lead to uncontrolled crypt formation and forming this adenoma. And it is believed that the mesenchymal cells are important in the cancer progressions. First, let's have a look on the stem cell niche on epithelial factors. Paneth cells, which found in the small intestine, they produce Wnt3A, Wnt11, EGF, TGF-alpha, and delta-like 4, which is a Notch ligand. This paper showed that when they sort the Paneth cells and LGL5 stem cell together as doublet and then put them in culture, they found that these doublets form colony with without win-free aid. However, with only LGR5 singlet, the colony forming efficiency was very low. However, when they add win-free aid back to the culture, you can see the more colony formed, and was comparable to the LGR5 Paneth cell doublet. So this experiment here shows that Paneth cells supported LGR5 intestinal cell culture by substituting the exogenous win-free aid. However, in colon, there's no Paneth cell, but this paper showed that at the bottom of colon crypts, there are c-KIT expressing cells. These cells express delta-like 1, delta-like 4, and EGF. However, they do not produce a win-free aid. When they co-culture these c-KIT expressing cells with LGR5 stem cells from small intestine and colon, they found that there are more organoids formed. After talking about the epithelial cell, I would like to talk about the mesenchymal fetus. This table here shows a summary of mesenchymal fetus found in intestine, and those labeled in red are some fetus that have been demonstrated to regulate crypt formation in vivo. And the last player in the stem cell niche is basement membrane. They are composed of extracellular matrix molecule, for example, type 4 collagen, laminin, and prenectin. They form scaffold for tissue organization and initiate intracellular signalings via integrin. And also, they sustain a local concentration of growth factors. Once again, my project is to study how mesenchymal fetus regulates the colon crypt formation. So the first question is whether we can create an in vitro models of epithelial and mesenchymal interaction. In this paper published in 2005, they show that when they isolate crypts from colon without mesenchyme, and then they put it back to the mouse as xenograft, sphere will form after 4 days. And after 14 days, new crypts will form in vivo. However, it is an in vivo model, and we are after an in vitro model. But this evidence here shows that it is possible to grow isolated crypt to form new crypts. So for an in vitro model that allows us to study crypt formation, we would like it to show crypt formation in vitro. There are high colony forming efficiency and early onset of crypt formation. Also, it is reasonably easy to follow by imaging. Here are some examples show what the in vitro model that we can use to culture epithelial cells from intestine. The first one is cell line, which is a 2D model layer of cells. Therefore, there's no crypt formation and no lineage progression was found in this model. And if we put the intact intestine into the culture, into a culture medium, there's crypt formation and lineage progression. However, it is very inconvenient to follow by imaging. And recently, there's an organoid culture developed where the crypts and mesenchyme were digested from intestine and then embedded in the matrix gel or other extracellular matrix. And in this culture, crypt formation were found from small intestine. And now I'm going to tell you more about this culture model. In 2009, Seattle set up this model where they embed the small intestinal crypts in matrix gel, and then put these 5 fetus into the medium. Here's how the organoids look like. And in our lab, we reproduce the result by start growing a crypt on day 0, and where this crypt form these organoids in the culture. And if we have a closer look on these small intestinal organoids, we can see there are 4 crypts, which are highly similar to the crypts in vivo. As you can see here, the Paneth cells found at the bottom of the crypts. And also, there's a lumen running on the apical surface to the central body here. However, as we want to study the colon crypts, so we asked whether we can use this small intestinal culture medium to culture colon crypts in vitro. However, the answer is no. When we put the colon crypts in the same culture medium, there's no organoid formation. And one of the explanations was maybe in the small intestinal crypts, there are Paneth cells which produce Win3A. And Win3A can enhance the organoid forming efficiency. However, in colon crypts, there's no Win3A producing cells. In 2011, the same group published another culture where they put Win3A into the culture medium, and they found that the organoid forming efficiency was much enhanced compared to the culture without Win. And here's an example of the organoids in the culture, where the green cells are chromogranin A expressing enteroendocrine cells, and MAC2 and the red cells are the MAC2 expressing globus cells. However, following the same recipe, by adding Win3A back to the culture, we only got this colonosphere, but not this organoid with bus. As you see, we got around 10% of the colonosphere form, which agree with the result of this paper. However, we don't get any organoids, colonoids, where it's a colonosphere with bus. Therefore, we think that it may be possible that, because in vivo, the colon crypts are surrounded by mesenchymal cells, and these cells produce lots of different cytokines and growth factors, and also extracellular matrix molecule that may regulate the crypt formation in vivo. However, in our in vitro culture, there's no mesenchymal support. And in our study, we are particularly interested in myofibroblasts, as it is the first cell layer that wraps around the colon crypt in vivo. These 2 examples here shows how the primary myofibroblasts co-culture with crypts from small intestine and colon, and to get in vitro crypt formation. In small intestine co-culture, we don't see a clear crypt formation. However, in this setting, we see bus form after 12 days in this co-culture. However, 12 days would be quite long for an experiment, and especially they are using primary myofibroblasts, which would suggest batch-to-batch difference. So we decided to isolate mesenchymal cells from the colon, from mouse colon, and to co-culture with the isolated crypts. The mesenchymal cells that we use is WEHI WEHI cells, which was isolated from the epithelial and mesenchymal cells by enzymatic digestion. So WEHI WEHI cell was isolated and characterized by Yumiko, and she finds that WEHI cell express alpha SMA, thymidine, COS2, but it doesn't express any epithelial cell marker. So we conclude that WEHI cells are myofibroblasts. And now we have a myofibroblast and colon crypt. So we co-culture these two together using a transwell setup. The WEHI cell were placed as a monolayer at the bottom of 24-well plate, and then we embed the colon crypt in mixed gel and put it in a mini cell insert with a 0.4 micron membrane, which are now the medium exchange between these two chambers. We found that with the support of WEHI cells, the crypt would grow into colonosphere and colonoid with bus, compared to the crypt-only culture, where only small colonosphere were found. And in this culture, not all the colonosphere would start to form colonoids, but some of them would just stay as a colonosphere. And here shows the colonosphere count. On day 1, we can see in the co-culture, WEHI cell support more colonospheres formed on day 1, which implies that it enhanced the survival of the isolated crypts on day 0. And we also count the colonoid number, and then we see in the co-culture more colonoid formed compared to the crypt-only culture. Now we have a co-culture that we can use myofibroblast WEHI cell to grow in vitro crypts from colon. And now we did some improvement on the culture. First, we scaled down from a transwell in 24-well plate to 96-well plate. And we also replaced the mixed-gel standard by mixed-gel growth factor reduce. As we found, using mixed-gel growth factor reduce, we can have a better colony forming efficiency. And we've also used purified Win38 and also outspawning through FC to replace the conditioned medium. Up to now, we can use the co-culture to culture crypts. Then the next question we want to see is whether we can use this co-culture to culture single cells from colon. So we use FAST to isolate epithelial cells from colon by EpCAM positive, size scatter low, and CD44 high. And then we subdivide the cells into 3 subsets, namely CD24 sublow, CD24 low, and CD24 medium. And then we put these subpopulations to culture with WEHI cells. And then we see the colony forming efficiency was highest in the CD24 medium subset. And here's an example of how the cell grow into a colonospheres. And in this culture, we also observe colonoid formation. So here's a brief summary of the co-culture. Myofibroblast WEHI cells enhance crypt survival, colonosphere growth, and crypt formation in the co-culture. And since the setup we use is transwell setup, there's no cell-cell contact between the myofibroblast and the colon crypts. So we conclude that cell-cell contact was not required in the WEHI mediated regulation. So it is possible that the WEHI cell produce some soluble factors that pass through the membrane in the insert to regulate the colon crypt culture. Therefore, we ask whether we can collect some WEHI conditioned medium and to see whether they support the culture. And since in the co-culture, the WEHI are under stimulation by the exogenous growth factors, so we ask whether this growth factor stimulation was required. In this experiment, which we grow the WEHI cells with or without the growth factors, and then collect the conditioned medium. We put the conditioned medium to the crypt culture, and then follow the colony formation. And we found that whether the WEHI cells stimulated with or without growth factors, there's not much difference in the the culture efficiency. And we should note that although there's the WEHI condition medium weakly supported culture, it is not as good as the co-culture, as shown as the dotted line here. So we think that it may be possible that the concentration of the stimulating factors were too low in the condition medium. So whether we can concentrate this condition medium and then we would be able to support the colon crypt culture. So we use ultrafiltration to concentrate the molecule of our interest in the retentate. And those small molecules will go to the filtrate. So we made the condition medium. We use YM3 central prep, which has a membrane with 3 kilodalton molecular weight cutoff. And then we concentrate the condition medium in retentate and put the retentate into the crypt culture and compare to the culture with WEHI condition medium. We made the condition medium after 24, 48, and 72-hour incubation. We can see after concentrations, the culture efficiency in terms of forming colonosphere or colonoid was much higher compared to the condition medium without concentration. And after several optimization, we produced serum-free WEHI condition medium, which they can support the culture fairly well compared to the co-culture. And serum-free is a very important property for the later purification step. And before I tell you more about the purification, I would like to tell you how these 4 exogenous factors affect the crypt formation in vitro. And our strategy is to take in each of these factors at a time to see their effect on colonoid formation. We found that when we took out Win3A and Alspondin-2, the colonospheres and colonoid was much reduced. Which suggests that Win3A and Alspondin-2 are necessary for colonoid formation. And since the experiment was done in co-culture, which means WEHI cells could not substitute exogenous Win3A and Alspondin-2. And this result was confirmed by this immunoblots here. It shows the WEHI cell do not express Alspondin-2 and Win3A. We've also used beta-catenin stabilization assay using L cells to look at the activation of Win signaling pathway. We see when Win3A was added, beta-catenin was stabilized compared to the control. However, when the condition medium was added, beta-catenin was not stabilized, which means that WEHI condition medium did not activate canonical Win signaling pathway. And for EGF and Nogan, we found that when they are taken out, there are more colonoid formation compared to the control. However, in terms of total colony formation, there's not much difference. And for this Roche kinase inhibitor, Y27632, which inhibits the epithelial cell from apoptosis after they detach from the basement membrane, we found that if we took this out, the total colony formation was reduced. And since this experiment was done in co-culture, we do not know whether the enhance in colonoid formation by taking out EGF and Ligand was a direct effect on epithelial cells, or is an indirect effect on WEHI cells. Therefore, we repeat this experiment using WEHI condition medium instead of the WEHI cells. And similarly, we found that when we took out EGF, Ligand, or both of them together, there are more colonoids formed, which confirmed that the effects we see in enhanced colonoid formation was a direct effect on epithelial cells. Now, just to remind you, the ligand is a BMP4 antagonist which bind to BMP4 and prevent them from binding the receptor. And when we talk about ligand, we see more organoid formation, which means more crypt formation in vitro. Therefore, we would like to add exogenous BMP4 to the culture and to see what happens. We found that when we add BMP4 to the culture, it inhibits the colonoid formation. As you see this one, there's the untreated control. And if we add BMP4, you see there's less colonoid formed in the culture. Also, we look at the TGF-beta signaling by adding TGF-beta receptor inhibitor. We added two different types of inhibitor at different concentration, and we found that when TGF-beta signaling was inhibited, there are more crypts formation in vitro. As you see, there are more colonoid formed. So now we know Wnt signaling, BMP, and TGF-beta signaling are involved in crypt formation. It's possible that they have crosstalk with each other. And from this experiment, we can see TGF-beta and BMP inhibits the Wnt signaling. In this experiment, they use human colon crypt culture and add TGF-beta to the culture. And then they found that when they add TGF-beta, SN2 expression was suppressed. SN2 is a gene of the canonical Wnt signaling pathway. And this effect could be rescued by adding the TGF-beta inhibitor. And if only the TGF-beta inhibitor was added alone in the culture, we see the Wnt activity was enhanced. And similarly, when BMP4 was added to the culture, the expression of SN2 was abolished. And when the ligand, which is the BMP4 antagonist, adds together with BMP4, we see the Wnt activity was rescued. And if only ligand added, there are higher Wnt activity compared to the control. Therefore, it is possible that there's crosstalk between the Wnt signaling and TGF-beta signaling in the colon crypts. And also with the with BMP signaling. And this signaling may crosstalk and regulate the crypt formation by regulating the cell proliferation. However, we don't know what's the relationship between the crypt proliferation, cell proliferation, and crypt formation. And so we add a DNA replication inhibitor, mitomycin C, to the organoid crypt culture and to see what's the effect on the crypt formation. Here shows the untreated control. As you see on day 3 to day 5, this colonosphere start to form buds in vitro. However, when we treated the culture with 10 micromolar mitomycin C, this colonosphere do not bud, but stay as colonosphere. And then we stain this culture using the standard EDU in this culture. EDU positive cells represents the proliferating cells. Here shows as a different stack in the 3D organoids. Here shows is the untreated control. And the right arrow here points to the proliferating cells in the crypts. However, when we treated with mitomycin C, the proliferating cells was much decreased. As you see, it's just a fail here, and there's no proliferating cells in 20 micromolar treatment. So to summarize, myofibroblasts with WEHI2 cells did not activate the canonical Wnt signaling pathway. And canonical Wnt, EGF, BMP, and TGF-beta signaling regulated crypt formation, partially by regulating the cell proliferation. After telling too much about the lung factor, I'd like to talk about the WEHI2 condition medium, which contain unknown colonoid stimulating factors. And we've done functional and biochemical characterization on these factors. For functional characterization, we determine whether these colonoid stimulating factors will potentiate canonical Wnt signaling pathway in the presence of Wnt3A. We also look at whether it can activate Notch signaling by looking at the induction of Notch intracellular domain, and to see whether WEHI2 condition medium is required before budding or during budding. Again, by using beta-catenin stabilization assay, you can see the Wnt3A could stabilize beta-catenin. However, when we add condition medium on top of Wnt3A, we didn't see an enhancement in beta-catenin stabilization, which suggested that the condition medium did not potentiate the Wnt activity, even when Wnt is present. And we also look at the Notch activity. As in this paper, they found that endothelial cells will produce a soluble form of JAK1, which is a Notch ligand, and it will promote the colorectal cancer stem cell phenotype. So we're interested to see whether in our myofibroblasts they will activate the Notch signaling. By using HCT116 human colon carcinoma cell line, we found that the WEHI2 condition medium did not induce the NICD compared to the EDTA positive control, which means that the condition medium did not activate Notch signaling. And now, let's have a look on how the WEHI2 condition medium induced budding. In our culture, we divide the culture into two different phases. The first 4 days called spheroid phase, where the colon crypts would run up to form colonal spheres and increase in size. And then until day 4, they will start to form buds, where we call it budding phase. In this experiment, we took out condition medium from day 4 and day 2, and to see how it would affect the crypt formation in vitro. We found that if we took out the condition medium on from day 4, which is the red line here, we do see this more colonoid formed compared to the negative control. However, if we took out the condition medium on day 2, we didn't see any enhancement in the colonoid formation, which suggested that the WEHI2 condition medium could induce colonoid formation when added on the first 4 days in the spheroid phase. And similarly, we do the other experiment and ask the question whether WEHI2 condition medium is required for budding in the spheroid phase. So we took out condition medium on day 2 and day 4. We found that even we took out condition medium on day 2 and day 4, there are more colonoid formed compared to the negative control. So these 2 experiments here conclude that whether we add the condition medium in the spheroid phase or in the budding phase, crypts will still form in vitro. Now I'm going to tell you about the biochemical characterization of colonoid stimulating factors. Here shows by adding the concentrated condition medium, it supports the colony formation compared to the negative control. However, when we treat the condition medium at 68 degrees for an hour, which would denature protein, the activity to support the colony formation was lost. So it suggests that the colonoid stimulating factors are proteins. And now we know the factors are protein, and then we want to separate the purify the factors by using gel filtration according to the molecular weight of the protein. And here's the elution profile of the gel filtration. And then we put each of these fractions in and resolve each of these fractions by using SS page, and we see the large proteins are elute in the early fashion, followed by the small proteins in the later fashions. And we put these fractions in culture. We found that these two red bars here, corresponding to the two peaks, could support colony formation, especially the colonosphere formation. And the purple bar here represents fraction 17, where you see that there seems nothing in the induction profile. However, if I have a closer look on the colonoid formation, where is the crypt formed, we don't see much activities in fraction 8 and fraction 13, which belongs to these two peaks. However, we do see an activity in fraction 17 and 18 compared to the negative control. And this fraction were found around 2 kilodalton to 17 kilodalton. Now we know that fraction 17 and 18 contains the colonoid stimulating factors. We use these fractions as a sample and biotinate the sample, and then using this biotinated sample, put onto the antibody microarray for the identification of the unknown factors. By using chemiluminescence detection. And this detection and this experiment we are going to do very soon. And by using this microarray called Weibo L series, we are able to detect more than 300 proteins in the active fractions. To summarize my presentation, we have established a co-culture using myofibroblast WEHI 2 cells to culture colon crypts. And we see colonoid formation, which is the crypt formation in vitro. And it was enhanced by the soluble factors derived from the myofibroblast WEHI 2 cells. We also found that canonical Wnt, BMP, TGF-beta, EGF signaling pathways were involved in the crypt formation in vitro. And lastly, small proteins smaller than 20 kilodalton were purified as colonoid stimulating factors. And that's the end of my presentation. And I would like to take this chance to thank my supervisors, Tony and Jonathan, for their support and giving me directions on my project. And also John and Jane for them to be my thesis committee. And also my colleagues in Burgess Lab, especially Chinwei and Yumiko, they gave me a lot of technical support. And Nicole, she did a very amazing work with the protein purification. And Janet and Nadia for them to give me some of the reagents for me to grow the colon crypts. And also the FAST Lab and my scholarship from the University of Melbourne. Thank you.
No, thank you very much. Please.
Great talk. I presume that all the work is done with mouse crypts, right?
Yes.
So the microarray, the antibody microarray, I think it's an amazing idea. Is it mouse specific or human specific?
As a mouse-specific microarray.
So I can't remember what your muscle gel looked like from the different fractions, but is there nothing you would want to attend mass spectrometry on?
Yes, actually, we plan to put the active fractions in the using the antibody microarray to test those proteins, and also we would try to put these active fractions to do a mass spectrometry and compare whatever comes up in these two experiments.
I guess in that same vein, I think when you were first investigating the colonoid stimulating factors from the conditioned media, it looked like you might have got an increased colonoid formation after stimulating the YH2 cells. Is that statistically significant, do you think?
Yes, there's a
left-hand side.
yes, there's a little bit increase in the colonoid forming efficiency, but but since we want to make a conditioned medium, and we're not sure adding these growth factors when we make the conditioned medium, it may make the system more complicated. So we decided to first just look at pure YH2 factors without any stimulation. And in our experiments, we showed that actually after we concentrate, we do see those factors exist even without growth factor stimulation. So we just proceed using without any stimulation. Yeah.
I think it's also fair to say that the YH2 cells like those growth factors, so after 48 hours, there's a fair few more of them in the cultures that produce whatever they're producing. So it may just be an indirect effect of the cell numbers.
So, of course, at the sort of level that you fractionated that conditioned medium so far, even finding out everything that's in your active fractions is going to give you a whole lot of things, right? So, how do you go from there to which one is actually responsible for the activity?
I guess first we use the antibody microarray and mass spectrometry to narrow down our candidate to see what will come up, and then we link back to what's reported in the literature may give us more information on how we choose what candidates to look further. And also we are going to further purify the active fractions with other column, for example, using ion exchange to get more purified fractions with different properties.
So if you look at the differentiation you put on your BNP or withdrawal experiments, the change differentiation as well in your cultures?
I don't have the answer. We haven't really looked at like the composition of the differentiated cells in the culture. No, I didn't.
One of the Yip K fractions had enhanced colonoid forming efficiency. I was just wondering, have you had a chance to look at LGR5 expression? Is it enriched, do you think?
I didn't look at the, after I shot the cells, I just put them in culture, but I didn't look at the LGR5 expression using, for example, real-time PCR. But there's a paper published in human colon, I think. They isolate the cell using Q24 and 44, and then they do a qPCR, and then they show that LGR5 was enriched in those fractions. But in my experiments, I didn't do the experiment.
Just a generic question, because you looked at the major pathways, Wnt, Notch. Is there any evidence that hedgehog has a role in regulating crypt formation? And do you think that one of the hedgehogs, because I'm not sure what size they are, could be involved in?
not sure whether heterogeneity would affect the crypt formation, but it's been shown to regulate the cell position in the crypts. So my guess is it's possible that they play a role in the crypt formation.
Kelvin, I can't remember whether you said the antibody microarray was custom built or not, and whether it was specifically, or the antibodies were chosen because they recognized very small proteins.
This microarray is not the one that we use is a commercial one, so it's not that we customize with some candidate factors. And these proteins, they are not all very small size factors. Some of them are large, some of them are small. But hopefully it can give us a primary screen that have an idea what kind of proteins may exist in the factions.
Kelvin, you've covered a lot of territory from that initial notion that maybe the niche cells do influence the epithelial stem cells to actually growing them in culture, and hopefully you'll get an idea of what molecules really do stimulate the process. But you've given us a very nice seminar today, and thank you very much on behalf of the audience.